We're going to further talk about muscles and get up into a process of explaining how muscles contract. And I'm going to tell you this right now, and then I'll tell you this at 6:40 or 6:30 or 6:15, whenever we finish. You have a homework assignment for Thursday. I never give you homework. I'm not collecting it. I'm not checking if you do it. But I want you to write this phrase down somewhere in your notes. It's called sliding filament theory. Sliding filament theory. And before you come to lecture on Thursday, I want you to pick 3 videos, your choice, on YouTube that you have watched and that you have, because Stacy comes in and she says, I'm very visual, and I know that a lot of you are very visual as well. So I'm going to be drawing a lot of things, which I think helps, but a lot of you get into the animations and the videos, and that can be very helpful. So watch 3, don't study, don't take notes, but watch 3, and then maybe when you hear some of the lingo, some of the vocabulary that I talked about, it'll kind of bring back a memory of what you watched. So that's the way I attack on something really challenging, and that's what I think Thursday's lecture will be. But it's going to be short, but it's going to be short and very sweet. So that's where we're headed. So what we're going to do today is get into the details of skeletal muscle. We're going to spend probably 20 minutes talking just about that model in lab. So that could be good for those of you who have the lab test tonight, and it will be helpful for those who have it on Thursday. We said that this next test, which is July 18th, next Thursday, is a test that's primarily dealing with skeletal muscle. We know that there's cardiac and smooth, but we talked about the fact that that's more of a 152 or a second semester kind of challenge. So we're going to say that this test is going to be mainly skeletal. I might have, out of 60, 70 questions, there might be 1 dealing with cardiac and 1 dealing with smooth, but that's it. So it's mainly skeletal muscle. One of the things that we're going to do once we understand the anatomy of the skeletal muscle is we're going to do some fun stuff. On Thursday, we're going to talk about the physiology of contraction, which is complicated. And then next Tuesday, our last lecture, it's going to be kind of like an exercise physiology lecture. We're going to talk about things like strength and endurance. That's a good one. Fatigue and things like that. So talk about why do you get that cramp when you work out or you exceed maybe what you wanted to do. So one of the things about skeletal muscle and muscle in general, both cardiac and smooth, is that it has these different characteristics. And the reason why I think that this slide is important is because this slide looks exactly like the slide that I'm going to show you after our next test, when we move into the world of the nervous system. Because nerves, look, are responsive. Nerves are conductive. Nerves, well, that's where we stop. So this is the bottom 3 is really what makes muscles unique. They can stretch, they can expand, get longer, they can contract and get smaller. But those top 2 are really important, and the physiology of responsiveness is kind of deep when we start talking about, how do we generate a brain signal? How does that communicate with a muscle cell? And that signal, it's electric. How does that travel? And what does it do to make things, happen on a molecular level? So the word conductive usually means something to do with electrical component. You hear about things being good conductors. When you think about maybe a severe thunderstorm and you say, oh my gosh, get away from that tree, it can conduct lightning or whatever. Something that conducts or allows electrical energy to pass through it is what we say is conductive. Muscle cells are conductive, which might be something new that you haven't really thought about. All of you know that when you look at skeletal muscle underneath the microscope, because we know that that's going to probably be on the lab test tonight and Thursday night, you're going to see a scope that looks just like this, right? You're going to see a slide with cells that have stripes, and then each cell has more than one nucleus. So if you see, and this is a hint, by the way, wink, wink. If you see a slide that has more than one nucleus, it should stop you right there and say, this is not cardiac, this is not smooth, it has to be skeletal. So the question, and again, look at that beautiful stripe or striations. We're going to begin to talk about what the striations are, but we'll go into great detail on Thursday. When we think about this whole idea of looking at a muscle cell and trying to explain why they have stripes and why they have more than one nucleus, one of the things you can see is that it all has to do about where they came from. Think about the last time we talked about something with more than one nucleus. It was an osteoclast. I don't know if you remember that. When we talked about bones, we said osteogenic cells make osteoblasts, which can become osteocytes, or osteogenic cells can multiply fuse together to form an osteoclast, and it took many osteogenic cells to make one osteoclast. It's the same thing here. So instead of calling them osteogenic cells, we call the stem cells myoblasts. And since it takes several myoblasts to form one skeletal muscle cell, we now know the reason why a skeletal muscle cell has more than one nucleus. That could be an NCLEX question. You know, I'm trying to get as much information as I can from my students. I just had a student, in fact, she was my tutor for 4 years, and you know, she kind of became part of the family in a way. And she drove up to Cleveland to go to my son's wedding, which I thought was awesome. And she just took the NCLEX on the 27th of June. And she passed it like in flying colors, which I knew she would, but I tried to say, okay, tell me some questions. She said, Mike, I think it's unethical. I said, just tell me some questions. You know? She said, whoa, well, I know there's a lot of questions dealing with bone physiology. What? Calcium and all that business we talked about. She talked about stem cells. She said there were 2 questions on her NCLEX test that dealt with stem cells. So when you hear the word stem cell, and you're going to hear it again next semester when we talk about how blood is formed in the bone marrow, just put a little star by that, because that could be something you see again in the future. We talked about the protective outer covering called the endomysium. And look at this. It says that even in us to this day, there are what we call kind of considered reservoir or reserve stem cells. And these stem cells that only produce skeletal muscle cells are called satellite cells. This is what we use to explain the rare situation when skeletal muscle cells replicate and repair. Because in most cases, they don't. In fact, what we see is that in most cases with skeletal muscle cells, oh, someone just texted me, hold on. Is lecture somewhere else today? I should say, yes, read the board. Because I wrote it on there.
Want me to go?
Yeah, no, yes.
What?
We're in O2, right? O2. Let's all guess, who do we think this is?
You're missing a lot of people. Yeah.
I put a sign on the board.
They all think we're.
I'm not gonna guess. You guys can.
We'll find out.
Oh, it was Ashley. Did you just text me? Oh, gosh. All right. So fibrosis, underline the word fibrosis. Fibrosis is what happens when there's damage to skeletal muscle cells. Instead of replacing them and repairing them, we have fibrosis, which is another name for scar tissue. Bodybuilders, people who want to get really gigantic muscles, will intentionally rip, tear, damage their skeletal muscle cells so that they can be replaced with much thicker, bigger scar tissue than muscle tissue. There you go. All right, so we're good with the connective tissue covering, I hope. Remember these, endo, peri, epi? Let's do a quick review. So when we look at a muscle, we said the outer component is made up of epi. We said each fascicle is surrounded by peri. If we take a fascicle out and look at one muscle fiber, one muscle cell, it's surrounded by endo. So it goes epi, peri, this is also peri, and then endo. Endo is around one muscle fiber. We talked about collagen being important, as well as elastic components, especially when we use the word tendon. So that protects from injury. And we're going to talk about some other elastic components today. Oh yeah, we're going to talk about some other elastic components today that allow for the muscle to stretch. Remember, elasticity is one of those kind of unique characteristics of muscle cells. I'm going to give you a second to whip it out and take a picture of this, because this one is money. This one is so important. So take a good picture of this. So notice the name that's on the top of the picture. It says muscle fiber. You could also write down in your notes that this is a muscle cell. You could also write down in your notes that this is called a myofiber. So myofiber, muscle cell, all refer to the same thing. So let's get an idea of what we're looking at. So this is not, notice, let's take a look, let's go back. This, what you just took a picture of is just this. Everybody good? Notice if you squint, it looks like there's fascicles in there. We don't call these fascicles. We call these groups of very tight proteins. We call these myofibrils. And that's what we mentioned about in lab. In lab, we didn't go into the details. We just said, oh, these myofibrils, these are the proteins. But today we get to go into detail. So let's take a look. This is one skeletal muscle cell. How do we know? Well, it has more than one nucleus. Look, 1, 2, 3, at least 3 nuclei, so that's good. If we look on the inside, we can see some very unusual things, but I want you to look at some of the names of the descriptive coverings and inner components. And notice, and write this word or prefix down, sarco. Wherever you see the word sarco, you're going to associate that with a muscle cell. So, for example, sometimes we call the plasma membrane, look, this outer covering, sometimes we call it the plasma lemma. But in a muscle cell, we call it the sarcolemma. Sometimes, or most of the times, we refer to the inner of a cell as the cytoplasm, but we're calling it the sarcoplasm. I mean, you're getting it, right? Instead of calling it the endoplasmic reticulum, which is one of those organelles that you had to remember, we call it the sarcoplasmic reticulum. So now, whenever you see that prefix sarco, you're thinking skeletal muscle cells. Okay, so we know that it's a skeletal muscle cell. It has more than one nucleus. It has these prefixes sarco. Let's talk about some of the other unusual things that make a skeletal muscle unique, right? I want you to write down in your notes that, yeah, every cell, mainly in the human body, has mitochondria. I can only think of a couple of examples of cells that don't have mitochondria, like red blood cells, for example. But most have mitochondria. But what's unique about a skeletal muscle cell is that they have gigantic mitochondria. And if you remember what the mitochondria are sometimes called in grade school, they're called the what? Powerhouse of the cell. They make the power, they make the energy, they make, ready, the ATP. I'm gonna say that again. The mitochondria are gigantic and they're responsible for making the ATP. ATP If that's not existing, then you're not gonna get muscles to contract. If you don't have the energy, that makes sense, you're not gonna contract muscles. So that's one of the ingredients for muscles to contract. ATP. Something else that you don't see and they don't show in this diagram, but in the sarcoplasm, we have two components that are really important. One is we have an oxygen binding component called myoglobin. So write down myoglobin. This is a protein that binds to oxygen. If you don't have oxygen, you're not going to get muscle contraction for a very long time. Muscles can contract without oxygen briefly, but if you do anything more than, let's say, 30 seconds, most of us start to breathe a little heavier, even if we're in good shape, which means we're trying to get more oxygen to our muscles.
What was that word again? I'm sorry.
Myoglobin, M-Y-O, globin.
Thank you.
And then the third substance that I want you to write that we would find inside the sarcoplasm is glycogen. Glycogen. If you remember back for test 1, we said glycogen was a polymer. And it's made up of many monomers of glucose, right? Glycogen is like the storage tank for glucose. So if we don't have glycogen or glucose, and if we don't have oxygen, and if we don't have ATP, we're not gonna get muscles to contract. So it makes sense that all three of those are found within the sarcoplasm. So you're gonna get a question probably on the test that will be asking you to explain the three ingredients, I guess, of the sarcoplasm that allows muscles to contract. You're gonna say oxygen and myoglobin, you're gonna say glycogen, and you're gonna say humongous mitochondria, making lots and lots of ATP. Okay, let's get into this crazy business. This looks like kind of a mesh. This is the sarcoplasmic reticulum. And the sarcoplasmic reticulum does just what the endoplasmic reticulum does, but it's unusual in the muscle in that it kind of blends into these three pipes. Three. Ready? We call these three pipes the triad. Oh my gosh. There will be probably 5 questions on the next test dealing with the triad. So let's talk about this triad. The triad are 3 pipes. 2 of them are called cisterns, or terminal cistern A. Let's freeze right there. How many of you know what a cistern is? Anybody in here? Tell me.
Is it like a hole in the ground, basically, like a well?
Kind of, yeah, yeah. So, my wife and I built our house, and we built our house when my wife was pregnant with Zach, who just got married. So we've been in that house for like 25 years. And when we were building the house, the builder, and this is in Colerain Township, this is not like out in east-west Claremont or something crazy. This is in Colerain Township. The builder said, there's no water where you want to build your house. So I said, well, I don't care. You figure it out. You're the builder. And he said, it's too expensive to bring water from like the road back to your house. So you have an option of a well or a cistern. And I looked at him and I said, this is not Little House on the Prairie. This is, this is 20,001, 2001, whatever it was. And it was in the 90s, 97, I think. And he said, that's all we can do. So we chose the cistern route. Now think about this. My house right now has a cistern, which is a big, gigantic cement tank that holds about 20,000 gallons of water. And we collect it, this is so barbaric, we collect it from the roof. It comes from our roof, through the gutters, into this filtering business, into this big tank, then it gets pumped back to our house. And that's what we've used for every day that we've been in that house for 25 years. And it stinks. I hate it. I look at the weather forecast and I look for thunderbolts. I don't look for sunshine, because if we are low in water, I have to buy water. I have to have like a water truck come to my house and dump water into our tank. So our kids are like, Dad, I love going to hotels. I love going to grandma's house because we can take really long showers. Yeah? Because it's like a 3-minute rule at our house. You gotta get in and get out. I mean, 5 people in our family. You know? So when I say cistern, ready? I want you to think of my house. And that big storage tank for water. But in mussels, the cistern is a storage, and there's 2 of them, is a storage tank, here we go, for calcium. The cisterns are storage tanks for calcium. You'll never forget it. Yes, Deb?
We have one.
You have one?
Yep.
Where do you live?
We're like, you know where Rumpke is?
Yes.
Right by Rumpke.
Well, that's where I live, in
Oh,
Corey
yeah?
Township.
Yeah.
What street do you live on?
We're right off of Crest on Rolling Acres.
Okay. You probably buy water from Toppers or something?
Well, sometimes when we would run out, it was back years ago where we actually found out there was a leak. We were
Yeah.
losing water, so we would hook up to the neighbors and then, you know, pay them for the water. But, we actually had to empty it, and we actually went down in there after it was all emptied, and, you know, they were filling
Repaired
out the
it.
cracks and stuff. It was really cool.
Well-
All the cement, yeah.
It's not good.
It
You have a sister?
Yeah.
Where do you live?
Georgetown, Ohio?
Okay, that's pretty rural. And so you have the short shower kind of rule too? Very nice, Dad.
Water guy.
The neighbor's the water guy, that's a good hookup.
I'm not feeling
Yeah, Michael?
So could that storage of calcium be what causes, to an extent, FOP?
FOP, no, that's a good question. Remember, FOP is when you have a situation where you have unwanted calcium. I'm thinking FOP probably has something to do with out of control osteoblasts, taking calcium from bone and building it. But that's a good question, but I don't think it has to do with this. So we have 3 pipes, 2 cisterns carrying calcium. Then we have something called the transverse tubule, or we'll just call it the T-tubule. And what I want you to write down in your notes about the T-tubule is that it is a conductive pipe, which means it's a pipe for electricity. So now we know what the cistern the triad consists of, 2 cisterns and a T-tubule. And you can see that there's triads everywhere. There's triads everywhere. And notice that the T-tubule is actually something that sticks up and out of the sarcolemma. So we now know a little bit about this crazy looking thing. Some of you remember studying the model in lab and seeing a bunch of stripes on the inside of that model. And I just said, don't worry about its proteins. But you can see that this dark and light and this dark and this light kind of banding kind of gives us the appearance of the stripes that we see underneath the microscope. So in order to understand these stripes, we have to look at these bundles, ready, of myofilaments. I'll write that word down. So myofilaments. There's 2 types. There's thick myofilaments, and then there's thin myofilaments. Thick and thin. And we say that a myofilament, and here you can see them down here, look, are encased or in groups called myofibrils. There's a lot of lingo here. So let's do it all one more time, because this is so important. If I give you this diagram and say, tell me what makes this muscle cell special, unique, you're going to start off by saying that there's more than one nucleus. You're going to talk about the prefix sarco. You're going to talk about the sarcoplasm and talk about myoglobin, glycogen, and these really large mitochondria. You're going to talk about the triads. and the two cisterns and the T-tubule. And then, of course, you're gonna talk about these bundles of myofilaments called myofibrils. If you can do that, you're good. You're good. All right, so there's a gigantic table. We'll go through all of this in a minute. You can see again, when we look at the cell has many, many nuclei. We talked about why we have more than one, because we said it takes multiple stem cells during development. We talked about the sarcolemma being the plasma membrane, having places where those T-tubules actually stick out of it. And we called the T-tubules conductive pipes, or tubes that carry an electrical current. So far, so good. Again, you can see that T-tubule right in the middle of the triad. We talked about sarcos, and we should be good with that. We talked about glycogen for energy and myoglobin for binding to oxygen. We said that the sarcoplasmic reticulum is something that fuses with this triad, blends into one T-tubule and two cisterns that store calcium. Yeah.
Are the triads random, or are they
Yeah,
just?
I think that, I don't know about random, but they're everywhere. So maybe they are. I'm not sure about their arrangement. That's a good question. So let's talk now about the myofilaments. Remember, a myofilament, let's go back. A myofilament is either a thick or a thin. I use two different colors to talk about this. We're gonna start with a thick myofilament, and it's called myosin. So the thick myofilament is called myosin. Something you need to remember. I would write down in your notes that myosin reminds me of a golf club. A golf club. So you can see that it has this kind of what we call this long polypeptide kind of tail and then the head of the golf club. And you can see the arrangement in a muscle cell. This myosin molecule is really hundreds, if not thousands, of these golf clubs with their heads sticking up. Their heads are going to be important for Thursday's talk. Then we get into something, and this gives you more of a perspective of the arrangement of these myosin molecules, is if you just focus in on this big, thick tube right here, you can see that some of these heads of the golf clubs are leaning to the left on this side. But then this area is what we call the bare zone. Look at this. There's no golf club heads in this area. And you can write down in your notes, this might be helpful, is that the bare zone is kind of like the transition from leaning to the opposite side or going to the opposite direction. So these are leaning to the left, and over here they're leaning to the right. So there's no heads in this bare zone. It's like the transition of changing kind of directions. Now if you squint in between these myosin molecules, you can see some red structures that look kind of crazy. And this is the second myofilament, and we call this the thin myofilament. So I want to draw this. I think that this one's worthy of drawing. So we're going to draw what what actin looks like. And every time I draw it, I think back to when my daughter was like in 7th grade.